And so remember the basic premise, the basic rule that we set forth before. Most all digestion and absorption occurs in the intestine. And as you look at the graphic here, you can see some of the various substances. I sort of summarized them here. Approximately, there's a general preference for the carbohydrates, lipids, amino acids, and some of the ions such as calcium and iron. Whereas distally, you're going to find B12, bile, various other ions that are absorbed. And this little diagram sort of takes you through some of the substances as they're absorbed at various points in the intestine. Now, the glands in the stomach are called pits. In the intestine, they're called crypts, C-R-Y-P-T-S. And the full name is crypts of Leverkusen. And so do not confuse your pits and your crypts, but additionally, make sure that as we continue our discussion, that you don't confuse intestinal structures with gastric structures. That's a very common mistake. I have no difficulty tripping up somebody on a test question confusing those two locations. By the way, this would be a villus here, sort of idealized villus. And so note that in the mucosa of that villus, as we saw before, there's a lamina propria. And in that lamina propria are going to be the intestinal capillaries that are responsible for taking up the digested, I wish we had time to talk about digestion in these cells, but taking up the digested molecules. And then that green thing is the lacteal. That's a lymphatic structure that also enhances uptake of the fats, assimilated fats from the GI tract. And of course, if the intestine is responsible for all these wonderful actions, it too is copiously secreting rather large amounts of, for example, bicarbonate to neutralize the gastric juice, a variety of digestive enzymes. And we're going to be talking about these in just a moment, but there are two classes of intestinal enzyme. There are pancreatic enzymes that are released via the pancreatic duct into the duodenum. And most intestinal digestion is achieved by those pancreatic enzymes. But their activity is supplemented by brush border enzymes that live in the microvilli of the intestine. And we'll return to that topic in just a moment. But also bile is released from the liver into the duodenum. A lot of mucus and some endocrine agents. For example, in that animation that we looked at previously, we saw reference to the intestinal phase of gastric motility. And remember, in that phase, substances released from the intestine travel through the blood and then targeted receptors in the stomach. So let's look at this very, very simplistic illustration and make sure we differentiate these two classes of enzymes. So everything in yellow in this graphic is a pancreatic enzyme. And everything that is green, there's not a lot of green here, is a intestinal enzyme. So in general, what the pancreatic enzymes are doing is all the heavy lifting. They're breaking up the carbohydrates by very powerful amylases. In fact, the amylases can reduce most of the carbs in the diet in a matter of, well, less than an hour in many cases. There are lipases that break down the fats, proteases for protein, exopeptidases that cleave off the ends of the peptides. And what the brush border enzymes in the intestine do are finish up that process. They clean things up. They take the products of the pancreatic digestion and then reduce those. They break down the disaccharides into monosaccharides. They break down the peptides into smaller, for example, dipeptides or tripeptides. That's usually what is taken into the blood. And then we need to talk about this one here called enterokinase or enteropeptidase, because it has a very special job. So getting back to this illustration, here are pancreatic enzymes. And one of the important pancreatic enzymes is something called trypsinogen. Now, what do we remember about OGEN? It's preliminary, it's inactive, all right? But this enzyme, enterokinase or enteropeptidase, acts upon it to render it active. It turns trypsinogen into trypsin. So the enteropeptidase is responsible for activating trypsinogen into trypsin. And then as you look at the bottom of our illustration here, what does the trypsin do? It activates all of these other pancreatic enzymes. Remember, all these are pancreatic. So the chymotrypsinogen, again, we talked about pro-hormones in the endocrine unit, propolipase, prophospholipase. I think you can probably infer what these things do from their names, but the trypsin activates them, and what rendered the trypsin active? The enterokinase, which is a brush border enzyme. So the brush border enzymes play a very important role, not only in cleaning up pancreatic digestion, but also in activating many of the pancreatic enzymes. Now we saw something like this before. We looked at something like this in the stomach. And this one's a little more complicated. It could apply to a variety of cells, but let's work through some of these transporters because you should be familiar with some of these if you aren't already. We've seen this before. Here's our good old buddy, the carbonic anhydrase reaction occurring in we'll call this the parietal, or we'll call this one of the typical cells of the intestinal crypt. And so notice again we have the two products, bicarbonate and acid, but pay attention to which way they go this time. So which way does the acid go following the carbonic anhydrase reaction? All right, it goes toward the blood. So the acid is leaving in exchange for sodium. So what would you call this type of transport? What would this type of transport be? Well, it would be more functionally. So what's moving, I guess what I'm asking is what's moving protons out? Yeah, the sodium ion concentration. It's really secondary active transport. Yeah, sodium potassium pump could be arguably an antiport, but it doesn't work the same way. All right, and here's our good old buddy, the sodium potassium pump maintaining the ion concentration gradients. And then also notice that the bicarbonate is being pumped where? Into the lumen. All right, so the bicarbonate is buffering the lumen of the intestinal tract. The acid is leaving. So pH should go up. All that is true. Now, in addition to those transporters, we should be familiar with most of those. We have this curious thing here. It's found in a variety of cells. It's present in renal cells we'll be talking about. It's a rather mysterious transporter in that it's moving 4 ions simultaneously in the same direction, called the NKCC transporter. The function, the mechanism is not well understood. In part, you can see that you have a couple of ions traveling with a concentration gradient, at least the potassium traveling against its concentration gradient. The usual explanation of this transporter is that it is acting to increase the osmotic gradient inside the cell. These transporters are thought to be important for regulating movement of water. So as you increase the number of particles inside the cell, that's going to increase the bulk movement of water across the membrane. Now, why is that important? Well, over here, this should look familiar to you. What is that? Bicarbonate is being exchanged for chloride. So we have that exchanger again. And then you can see the chloride then, in order to maintain this gradient, is more or less being transported out of the cell by this particular transporter, which also is present in many tissues, in the intestine, in the lungs, in the liver, and elsewhere. This is the CFTR channel, or the cystic fibrosis transmembrane conductance regulator.
All right.
And if it's doing its job properly, if that transporter is functioning properly, that is to say it is not mutated, and there are over 100 mutations of that particular transporter, chloride leaves, the sodium tends to follow the chloride, and what's going to follow the sodium? Water. And so this particular chloride transporter is very essential for maintaining the proper level of hydration in this case on the luminal side of the luminal side of the intestinal cell. If it doesn't function properly, water transport does not occur, and there can be aberrations in the formation of mucus. So as you look at the cell, just be familiar with the general organization of transporters and their general functions. We talked about a number of hormones. A couple of these you should be familiar with, the secretin and the CCK. Now remember, gastric emptying is going to be regulated with respect to three primary criteria: the composition of the chyme, the more fat and protein, the slower the emptying, the pH of the chyme, the lower the pH, the slower the emptying, and the osmolality of the chyme, the greater the higher concentration, the slower the emptying. And as we saw in that animation, there are a couple of hormones that are important for conveying signals about the composition, pH, and concentration of the chyme from the intestine to the stomach. Because remember, gastric emptying is going to be regulated by that caloric valve, that gastroduodenal junction. Secretin and CCK are both intestinal hormones that act to delay gastric emptying. Secretin generally secretion levels are increased when the pH drops below about 5 or 4.5. Again, telling the stomach, hey, back off, cut it back a notch because it's getting way too acidic in here. CCK does the same thing. It also acts to moderate gastric emptying, but it also does something else. It triggers release of bile into the duodenum. And the bile, I think we've talked about it before, acts as an emulsifier of fats. Helps to basically facilitates the ability of lipases to access the fat. So again, these are hormones. Just be sure you understand the difference between paracrine activators in the stomach and hormones. There are cells in the stomach and cells in the intestine that release compounds that act on neighboring cells to moderate their activity. But these are both intestinal hormones that travel with blood back to the stomach. All right, so that's the worst of it. Make sure you are familiar again with the transporters and understand the general mechanism of some of these hormones and how they alter gastric emptying. We've already talked about the liver. The liver is about the same mass as your brain. It's indispensable. You probably live longer. I think I've shown this to be the case. You probably live longer without your brain than you could without your liver. The liver, if it's gone completely, you're probably not going to survive for more than 45 minutes or so. And we've talked about its role in detoxifying, processing the portal blood coming from the GI tract. It regulates blood glucose levels through the depolymerization of glycogen, for example. Has a variety of mechanisms for releasing just a host of enzymes and regulatory proteins into the blood. I think there's some estimates that there are almost 1,000 different proteins that are released at various points in the life cycle from the liver. It's important for red blood cell recycling along with the spleen. It serves as a repository for a variety of ions and minerals, including, for example, iron, the vitamins A, D, B12, and so on. We've already pointed out and seen that the liver secretes bile that has been released into the duodenum, so very, very important organ. As part of its role in processing the portal blood, the blood that arises from the GI tract, you should be familiar with something called the first pass effect. And basically, anything that passes through the liver is going to get worked on. It's going to get probably reduced, chemically altered in some fashion. This is true for antibiotics. This is true for a variety of therapies that you'll probably be administering to your patients. A lot of therapies have to take into consideration the effects of the liver on the drug, the first pass effect, and basically have to be provided accordingly. Famous example of penicillin. The first case of penicillin, it was having miraculous effects on the patient. who I think suffered from, who had an infected rose thorn infection, and he looked up just awful. You can look at pictures of him, but again, they didn't account or didn't have enough of it to deal with the metabolism of the liver, and he expired after this miraculous initial recovery. So we talked about the anatomy. A lot of you probably are fond of liver loaf. No, I am. Liverwurst. This is just to remind you that in order for the liver to do all the wonderful things that it does, it needs both oxygenated blood supplied by the hepatic artery, and of course it's going to work on the hepatic portal blood, and all that's then going to be mixed together in the sinusoids. We talked about the lobules, and then conveyed to the inferior vena cava by means of the hepatic vein. So the bottom line is, don't confuse the hepatic vein with the hepatic portal vein. And so in the anatomy, we talked about the portal triad. You can see the mixture of the portal blood here with the oxygenated blood, and then the bile moving in the opposite direction into the portal triads of each lobule. All that blood is then collected in the central veins of each lobule, and as we saw in the previous graphic, all of that is going then to be delivered into the vena cava by means of the hepatic vein. The bile I mentioned a moment ago is most important for emulsification of dietary lipids, dietary fats. As you can see the illustration in the background there, that these are intended to be lipases. The lipases look like little Pac-Man going to work on the dietary fat. And so bile, like the detergent that you use to wash dishes each day, the surface active agent we talked about earlier, acts to break the big glob of fat that you see here into smaller and smaller units that the lipases can then go to work on and begin breaking down. So that's what we mean by emulsification. Emulsification is not digestion. We're not talking about any type of enzymatic response occurring in the emulsification by itself. Rather, there's just a mechanical reduction such that the enzymes can go to work on the smaller units of fat. Bile consists of a variety of components, mostly water. There are a lot of bile salts, so both steroid and amino acids, and then there are a variety of pigments resulting from the breakdown of red blood cells, and recycling of red blood cells. The gallbladder can hold up to a few mils. This, I was going to say, it usually holds about 60 mils. And there we go. And then there's a daily churning of a liter, liter and a half, because of the amount that's released in response to dietary lipids. The liver can become, I think we saw the fellow drinking in the other day, the fellow drinking the beer, the liver is very vulnerable to alcohol toxicity. One of the terminal conditions that can result from that type of toxicity is cirrhosis. Initially, the liver, in the fatty tissue in the liver increases. That's called steatosis. And as that steatosis, those fatty deposits give way to scar tissue and other non-productive tissue types, it turns into cirrhosis. And you can sort of see histologically the transition of normal parenchyma of the sinusoids of the liver into scar tissue and fatty tissue. So we've talked about the anatomy. We don't need to discuss haustra, omental appendices, diverticula. You're familiar with all of those terms, Taenia coli. We talked about the importance of rocking motility in the colon. One of the important functions of the colon is to extract water from the chyme, because of course the body wants to conserve as much water as possible. Downside of that is as the water is extracted from the chyme, it becomes more like cement. And thus, if that cement is not kneaded, I don't mean N-E-E-D, I mean K-N-E-A-D, it's not broken up like bread dough, it can set up and become very difficult. And of course, that's called constipation, right? That's one of the possible causes of constipation. So in the colon, there's going to be segmentation, except the segmentation in the colon is called haustration. The segmentation in the colon is called haustration, and it's achieved by those very powerful radial muscles kneading and breaking apart the chyme. The rocking motion, the rocking motility in the colon is achieved by both peristalsis and antiperistalsis going backward and forward, once again to avoid the chyme setting up. And of course, we talked about the migrating myoelectric complex. Well, eventually, all of that stuff that moves from the mouth, esophagus, stomach, and the intestine, it all ends up in the colon, and it often stays there for a while, especially with our modern lifestyle. But the clearing motility as it occurs in the colon is called a mass movement or bowel movement. That's usually what we mean by the clearing motility. So I've indicated some of the important functions of the colon, storage being prominent among them. Water reabsorption, retention of water, conservation of water. There is a very high volume of mucus released by the colon. We talked about the anal columns this morning. Haustration, segmentation to prevent constipation, at least minimize constipation. There is a important degree of bacterial metabolism. You can see some of the vitamins that are generated in the colon. About half of your vitamin K, which is important in blood coagulation. Much lesser amounts of B vitamins, all from your little intestinal buddies down there, and you have several pounds of bacteria, which, as I pointed out too, are probably important in altering your dietary preferences and perhaps even your behavior. To that end, even your mood. So notice these invaginations in the mucosa of the colon. You see lots of, there are no villi, so there are no villi here, but you often see lots of, you don't really see very clearly here, but lots of goblet cells. These goblet cells secrete mucus, and they're quite abundant in the colonic invaginations. We can see very clearly the haustra here, looking at the interior of the intestine. Both illustrations. We talked about refluxes again. Remember, I talked about the gastrocolic reflex. The admission of ingest into the stomach is going to cause some type of change in the motility of the colon.
The presence of food in the stomach and chyme in the duodenum stimulates mass movements in the colon. Mass movements are integrated by the enteric plexus. They propel the contents of the colon toward the rectum. The presence of feces in the rectum stimulates parasympathetic and local reflexes that result in defecation.
Which again, hopefully you have something to say about. You wouldn't want this to be exclusively a reflex response. I'm sure we can all agree on that. But in any case, as you go back to the illustration there, what if this occurs too rapidly? What would we call the condition in which that arrow is moving the chyme through too rapidly? Yeah, that would be diarrhea, and of course, if it doesn't occur sufficiently enough, that might be, among other possibilities, a manifestation of irritable bowel symptoms of some other similar condition. This is a discussion of IBD here. What you're going to find that IBD, like most of the diseases, most of the important disorders and pathologies that you will be dealing with, they have both a genetic component and an environmental component. So here the implication is that this patient suffering from IBD maybe began with a normal gut, then there was some sort of trigger. Environmental trigger. It could have been an infection, it could have been something they ate, it could have been a medication, it could have been an iatrogenic intervention. And as a consequence, depending upon a variety of factors, which we really don't know, we don't know why some people suffer from, for example, heart disease or diabetes in some cases, others do not. But depending upon the ability of the individual to tolerate the insult, they may either end up with the disease IBD or not. Again, combination of both genetic and environmental factors. All right, this morning we talked about the anal canal. All of this should look familiar to you. You can see here's our internal anal sphincter here. That's the reflex portion that the fellow was just talking about in the animation. This is smooth muscle for the most part. External to that is the external anal sphincter, and these work analogously to the internal and external urethral sphincters, in that both must relax in order for elimination to occur. Now, when we talk about elimination, of course, it's going to be highly variable. How often should bowel movements occur? Anybody have any strong feelings about that? Something you surely must have thought about. Once a day, once a week, once a year? I remember as a child going to visit the medical museum in Washington, D.C. I think it affected me and worked my brain thereafter. But as you walked into this medical museum, I don't think it exists, at least not in the form that it did then, there would be row after row after row after row of bizarre medical exhibits. They would have skulls and heads and organs and arms, elephantiasis, burned patients, mummies that were cleaved, and all that sorts of disarray. But what I still remember is a constipated large intestine in a glass case, glistening in the flickering fluorescent light. And it was fatal that the intestine was about as wide as it was long. It was quite remarkable. So clearly that individual, for whatever reason, did not have the adequate number of bowel movements. But you should be aware that your patients can have a broad range. It may be several times a day or perhaps only a few a week. Those would be relatively normal. And of course, if it is deferred, and all of you are probably in the active enterprise of deferring your bowel movements in deference to classes and so on, that can result in potentially constipation with adverse consequences. Hemorrhoids, we saw the hemorrhoids in the venous plexus this morning. Anal fissures, that's something we can all talk about at dinner tonight. I always like this illustration, sort of summarizing the effects of constipation. When that chyme sets up and is no longer moving at the appropriate rate. Of course, this is from the New England Medical Journal. What happened here? The New England Journal of Medicine. Saying you're a PA, this is what you're in for. What do? I've got a pain. I don't feel good. I've got a pain. Would you think to look for a beer bottle in the wrong spot? They had to go
That could have gotten there only one way.
Apparently did some surgery.
Yeah.
Sweet snacks, bowel cancer. There's just been a wealth of research again on the fact that now they call this the gut. What do they call this? The brain-gut enteric microbiota axis. And this is the axis involving the hormones and the effect of these hormones on your autonomic nervous system affecting your mood. There have been many studies with animals. For example, you take gut bacteria from obese mice and put them into the skinny mice, emaciated mice, and what happens? The skinny mice become less active and they start gaining weight. So was it the bacteria that caused it, or was something caused those bacteria to develop? Yeah.
Have they done studies on like positioning, like the squatty potty and all that?
Yeah, you'll find if you listen to any, I can think of several podcasts I listen to that advertise the squatty potty, and they will brag that they are the beneficiary of numerous scientific studies showing that it's beneficial. But if you actually look at some of the studies, they're not in very reputable journals, and a lot of them have not been repeated. And I think you probably know why. Anybody who wants to involve in a squatty potty test? It's very hard to get people to. You want to explain the squatty potty?
Lift your legs up and then you poop like a caveman.
Never gone camping? I was a child. All right. Any questions about the GI tract? Well, we need to get moving.
Thank you.
But you don't lift your things. They're forced. Yeah. I read that study.
Okay, let's talk a little bit about, let's conclude our physiology with a brief discussion of renal physiology. I always put this at the end because
It's your favorite.
it so effectively summarizes everything we've been talking about. It involves everything from osmosis to transporters to endocrine system, you name it. The renal system, the renal physiology has it. I think you're familiar with the anatomy. We don't need to talk too much about the anatomy. Basically, the distal portion of the kidneys on each side are roughly coincide with the 11th and 12th ribs. Going to be a lot of variation there depending upon a number of factors. We've already talked about the large volume of blood passing through the kidneys to serve the various functions that they perform. Notice in this illustration the renal veins are anterior to the renal arteries. All right, let's talk a little bit about the organization of the blood supply. There is an error on this illustration, but that should be no big problem. The reason we need to be familiar with the organization of blood vessels is because we need to understand the blood supply to the nephron, the functional unit of the kidney. So here we can see the renal artery coming into the hilum of the kidney. It's the large artery there that then branches, the renal artery branches into what are called segmental arteries. These are the segmental arteries you see here. So there would be a segmental artery. And then those segmental arteries, and this is where I think the error is in the picture here, those segmental arteries then branch into interlobar arteries. So I'm sort of highlighting the interlobar arteries there. Excuse me if I color a vein. Wouldn't know if I were coloring a vein. So the interlobar arteries tend to leave the renal pelvis there, the calyces, and pass up along the pyramids. These are the pyramids. This is the cortex out here. All of this is the medulla. The interlobar, again here it's called interlobular, but the interlobar artery, so that's incorrect. The interlobar then sort of arc around the pyramids, at which point they become arcuate arteries. Is that in there anywhere?
No.
Arcuate arteries. And then the arcuates give rise to the interlobulars, sometimes called cortical radiant. So again, that particular attribution is incorrect, but the spelling is correct. It's just do not confuse. Let me put these words in here. Do not confuse interlobar with interlobular. These are the smaller vessels. The interlobular vessels are also called in many medical texts cortical radiant arteries, because they're sort of radiating from the arcuates. But again, just be aware of this error. I think it's corrected in a later illustration we'll look at, but just be aware of that ambiguity. So these would be the radiant arteries out here, interlobular, whichever you prefer. Additionally, you can see that these pyramids are going to drain. Basically, this is where the urine is going to be formed by these pyramids. There's a papillary region down here. The papillary region of the pyramid then extends to become the calyx. Calyces is plural. There's a minor and major calyces. These up here are the minor calyces. Down here we would have, kind of hard to see here, inside, on the other side of that blood vessel would be the major calyx. And then the major calyces are going to empty into the renal pelvis, which is this large opening you see here. And then that will give rise to the ureter. Any questions about the organization of the blood vessels and the passage of the urine? Yes.
So you said on there that was wrong, right?
Yeah, this one is, well, actually the vein, the interlobular artery is misnamed. So that should be interlobar. And that will become important as we'll see later. And I do think there's a subsequent illustration like this in which the names are correct. And this cadaverous specimen here, you can again see the cortex out here. These are the renal pyramids, calyces, pelvis. You can see the renal artery and vein at the hilum. Again, all of this is the hilum. And pyramid, column, cortex. And then all of this, approximately speaking, is going to be the medulla. No, in these cadaver preparations, you never know what you're going to get. I think all of this is just an artifact, but there's really technically only one pelvis. I don't know what you're looking at that V there. I don't.
Well, no, up above it, it's like almost two.
Oh, I see. I would say that the pelvis would be down here. These would be calyces. So I would call these calyces. And they have the papillary region draining into those. But certainly that pelvis would extend up into here as well, except for the artifact. So we've used the word hilum in many cases, but with reference to not only the kidney, but for example, the lung and some other tissues where all the neurovasculature and lymphatic structures pass into and out of the tissue. Let's talk a little bit about the functions of the kidney, because we won't have a lot of time to get into all of these aspects in great detail. The kidneys play a very important role in filtering your blood at a very high rate. At about any given time, about 20% of the blood in your body is passing through your kidneys. Filtering that blood and sort of processing that blood, much like the liver, but in a rather different way, processing that blood for agents and substances that need to be removed from the blood because of the possibility of toxic accumulation. Most of those metabolic wastes that can prove to be toxic are products of nitrogen metabolism. And so all of these substances that you see down here, labeled 1, 2, 3, and 4, these are all products of protein metabolism. They must be dispensed, excreted from the body in order to maintain appropriate nitrogen balance, because nitrogen can be so toxic, at least in its accreted or combined form, chemical form. Urea, you probably remember very simple, just NH2, ammonia, which you know how delightful that is to either drink or smell. We'll talk about some of these others. Other organisms actually excrete uric acid in greater volumes than urea. We release primarily urea. And then creatinine, we'll talk about later. Notice the spelling is very similar to a substance we spoke of with respect to muscle metabolism. And they're very similar in their composition, but there are different substances. So again, the big theme here, we talk about these metabolic wastes. They're not exclusively nitrogenous waste, but that's the primary species of waste that is discharged and regulated by the urine. At the same time, the kidneys are filtering, as I've indicated, your blood at an extremely rapid rate. And some of that stuff is some of the materials in the blood are being removed from the blood inappropriately. So the kidney, miraculously, has the ability to return the substances that it took out of the blood through a process we'll talk about later, but it basically ensures through various mechanisms that agents that are critical for vitality remain in the blood. Kidney is important for regulating body fluid compartment volumes. For example, if you prove that the kidneys are filtering your blood in such high volumes, what would happen to systemic blood pressure if we suddenly shut down renal function altogether? Let's just stop putting blood through the kidneys. What would happen to systemic blood pressure? Yeah, well, I see several of you pointing out it would increase dramatically. So that's one of several mechanisms in which, for example, blood pressure can be regulated by the kidneys. We'll talk more about that. Osmolarity can be regulated. Compartment osmolarity can be regulated by simply regulating the volume of electrolytes and particulates in the blood. We'll see how that is achieved. There are numerous transporters in the epithelial cells of the renal epithelium. It's kind of redundant. That are taking electrolytes into and out of the filtrate. We'll look at the importance of some of those. The endocrine is an active endocrine tissue. The kidney is an active endocrine tissue, for example, releasing renin, which by means of the RAAS pathway induces release of aldosterone. We've already looked at EPO. What is EPO? Erythropoietin, important for what purpose? Yeah, increasing the hematocrit, increasing the hemoglobin level, and so on. So that is a renal hormone. As time permits, we'll talk a little bit about acid-base regulation. You're going to have I think you'll have almost an entire semester devoted to acid-base regulation by the kidneys and the respiratory system. We talked a little bit about respiratory regulation of blood acidity. We should have time to talk a little bit about the ability of the kidneys to regulate pH levels, blood pH levels. The kidney, like the liver, is an important detoxifier. In fact, even if a potentially toxic substance is not filtered by the kidney can nevertheless has a mechanism for getting rid of it, and that can be quite remarkable. The downside to that is that, like the liver, sometimes the kidney will take substances from the blood and get rid of them. Substances that may be therapeutic, drugs, antibiotics, and so on. And there is some limited capacity for blood sugar regulation by the kidney, but in most cases, when we talk about renal gluconeogenesis, the patient's in pretty bad shape, at least nutritionally. So many functions. These next few slides you've seen before. This is just to remind you that you need to be familiar with the relationship between the ICF and the ECF. You need to go back and recall those ratios if necessary. We talked about the 24-60 rule. Can't tell you how many times the instructors of your more advanced classes complain that their students don't know the 20-40-60 rule, so make sure you know that. Make sure you understand the relative ratio and importance of these. We talked about the fact that the cells differ dramatically in their proportion of water. For example, muscle cells are going to have a much higher water proportion than would adipose tissue, which is inherently hydrophobic. And I don't think we looked at this before, but here's a little table showing how changes in osmolality can be altered by changes in volume. So with the exception of this one, to get us in a greater discussion that we need to get into, you should be able to explain each of these little boxes here. For example, how would ingestion of hypertonic saline increase blood volume and increase osmolality? Anybody explain that? First of all, where are you going to get your hypertonic saline? I guess you could make it. You're going to, so hypertonic saline means what? That it has a higher concentration than, substantially higher than this, and you're going to be drinking it presumably, or injecting it in sufficient quantities to both increase volume and osmolality. Somebody explained to me how simple sweating, how simple profuse perspiration would increase osmolality but reduce TBW? Anybody want to give it a go? When you
The body can change how concentrates the sweat is, so you lose more water than salt, so your remaining body water has more salt, higher concentration of salt.
right, if there's greater proportionate loss of water to electrolytes, then that's going to increase the osmolality. At the same time, you are sweating, you are losing, presumably both. So that's going to decrease the volume. So work through each of these boxes and make sure, for example, you see how drinking a large amount of pure water, that's going to decrease osmolality and increase volume. Should all make sense to you. And of course, the graph here, we're looking at fluid intake. Basically, this is a little bit misleading. The implication is you can drink as much as you want. It's not going to alter your blood volume. Why is that? Why, if I see you down at the bar this weekend and you're taking it in pretty regular, why would that not? Why would that not be a problem? What's going to happen is
Peeing it out.
you're going to start. Hopefully, you're going to be peeing more as well. But what's the problem over here? What's indicated by hypovolemia and death? Right. Here we have insufficient intake of fluid. Could be loss of fluid by any number of pathological processes, or there could be irreversible dehydration. Okay. So we didn't get very far there, but next time then we'll begin a discussion of the nephron. So I've posted two presentations on the kidney. I may or may not post a third, or I